Hi, welcome to today's practical class. For our learning purposes today, we're talking about malaria parasite test. Now, malaria parasite test is carried out to identify various species of plasmodium that affects the human person. Now, it's either done in a thick film or in thin film. Well, we will concentrate on thick film today. Thick film is used for parasite count, but thin film is used for speciation. That is, when you're trying to identify what species of plasmodium is actually infecting a human. So for today's purposes, we'll be learning on how to make thick film, stain, and count. So that's what we're going to do today. Later, we'll learn subsequently how to use thin film for speciation to know the different species. And we have four different species of plasmodium. We have Plasmodium falciparum, which is the most prevalent in sub-Saharan Africa. Then we have Plasmodium ovale, we have Plasmodium malariae, we have Plasmodium vivax. Now, it's important to know what species is affecting the human because different species is what they actually cause. For example, it is popularly known that Plasmodium falciparum causes what is known as sequestration. What do we mean by sequestration? It means that Plasmodium falciparum has the ability to sequester under tissues and organs of the body in such fashion that even after treatment, if not properly done, will come up again as a reinfection. So it has the ability to sequester, that is to hide under organs and tissues of the body. So falciparum has that capacity. We have vivax and ovale, which where we hear about relapse. Relapse occur when there is a particular step of the malaria parasite known as hypnozoans. Now, hypnozoans are found in the liver. Now, at that stage, some of the hypnozoans do not progress to infect the red blood cells in the erythrocytic stage of malaria parasite infection. It does not go to the stage, it remains latent in the liver. And after treatment, begins again in the cycle to cause a relapse. So Plasmodium ovale and Plasmodium vivax are known to do that. So it's important we know what species is infecting the human. But for today's purposes, we are going to learn how to use thick film to identify malaria parasites. And you can only do count with the thick film. So that is what we are going to do today. So for our learning purposes today, we have our blood in our EDTA container. We have already prepared Giemsa working solution, 10%. Um, Depending on your choice, the percentage depends, the length of stain depends on your dilution factor. But standardly, we have 1 in 10 dilution factor where you add a mil of ginger stock to 9 mils of distilled water. And then we have already labeled clean grease-free slide, labeled sample B. We have immersion oil for viewing. So we get on with it. So onto a clean grease-free slide, you make a thick film about 2 cm in diameter. Before you do that, you want to mix your blood sample properly. You don't want a situation where the samples are already packed, the cells are packed down. So for every test, there is a need to rock or mix properly before you obtain a little quantity of it. So you obtain a little quantity of the blood, about a drop, and you make your smear. And like I said, the smear should be about 2 cm in diameter. there are bubbles, you remove them to obtain a good smell. So your smell should be 2 cm in diameter, and you should create room for labeling, and the smell should not come to the both end of the slide. You After making the smell, the thick film, this is a thick film. Now make sure your smell, your film is readable through a print. What do we mean? Okay, now there is some write up on this marker. You should be able to read what is on the marker from this thick film. So we're going to place it on the thick film now and see if we can read. Okay, I can actually read from this. Okay, recap after use. So you see that your smell should not be too thick or too thin. So you should make to ensure that you have made a good thick film. It should be readable through a print. So we're going to allow this to dry before we can continue with other staining processes. Yes, our film is all dried. Now, this is how your thick film when dried should look like. This is how it should look like when dried. You see, this is how it should look like when dried. So now we're going to go on to staining with our working solution. I forgot to mention that for the Giemsa working solution, since it is photolabile in nature, that is, it is sensitive to light, such as sunlight. So for every working day, like you come to work in the morning, you have to prepare fresh working solution to ensure that your Giemsa working solution is still active. So we just prepare this 10% working solution, Giemsa working solution now, because we don't want to use a working solution that has been degraded due to light. So we go on to stain. We flood with the Giemsa stain for 10 minutes because we are using 10%. If you're using 20% Giemsa working solution, you stain for 20 minutes. So depending on your dilution factor, you start your timer. And don't forget, while staining, if there is any bubble, ensure to always remove to prevent the smell from being wrongly stained. So I'm going to remove the air bubbles now, because I don't want some parts to not pick the stain. All right, we're already timing 10 minutes. Right, it's 10 minutes already. So we're going to rinse off the Giemsa stain. Now I like rinsing really slowly using my beaker. I don't like using under flowing waters to avoid washing off of the stain. So I like rinsing. You see, gently to avoid washing off the film. Then you drip dry your blot. This is essence of labeling your slide, so you know where the stain, the smell is. So avoid cleaning off the smell. So you blot and you drip dry. And then you allow this to dry before we go on to we go over to microscopy. Yeah, before we go over to microscopy, we have to apply a drop of immersion oil, which facilitates viewing under the immersion oil objective lens. So, like we said in our previous video, when you're viewing for a stain slide, you rack your condenser up, you open the iris diaphragm, and the stain slide, the oil of the stain slide must hit the lens. So in the next slide, you're going to see how the malaria parasite is supposed to look. You have full rings. You have full rings. This this is a typical trophozoite. I know my marker. This is a typical trophozoite. That is having a full ring. This is the chromatin dot, and this is the cytoplasm. Note, the cytoplasm will stain blue. Unlike other Romanowski stains, that the acid portion of the stain, which is the eosin, stains the basic portion of the cell, which is the cytoplasm. In Giemsa staining, the acidic portion of the stain, which is the eosin, stains the acidic portion of the cell, which is the nucleus. And that is the reason why the chromatin dot will stain red, because the chromatin dot is a part of the nucleus. So it's going to stain red, picking the color of the eosin. While the basic part of the stain, which is the methylene blue, will stain the cytoplasm, which is the basic portion of the cell. So for Giemsa stain, the acidic portion of the stain stains the acidic portion of the cell, while the basic portion of the stain, which is the methylene blue, stains the basic portion of the cell, which is the cytoplasm. Don't forget to actually ascertain that this truly is not an artifact. I must see a clear vacuum. There should always be a clear vacuum in the trophozoite. So this is a typical trophozoite of Plasmodium. Now there are other shapes you want to look out for. You can see shapes that are not actually full ring like this. You can see shapes like this, or you can see shapes like this, or you can see shapes like this. So at the times you only see the chromatin dots, depending on factors such as if such patients have already commenced treatment. So these are factors that can affect if factors that affect if you see full ring or other forms such as this. So as we're going on to our microscopy, these are forms we are going to look out for while doing our microscopy. And note that for thick film, you are not going to see the plasmodium species inside the red cells, because during staining it has been lysed. So for thick film, you don't see the plasmodium species inside the red cell anymore, but it is seen open because during staining, water lyses the red cell to expose the plasmodium. So you see why thick film is for count. Then for thin film, it is not, the red cells are not light because they are retained using methanol, which acts as a form of physitis. Hence, you see the plasmodium species inside the red cell. So that is basically the difference between thick and thin film. So this is what you should look out for. Now we're going to go over to this thing. We already have our immersion oil on top of our stain slide.